my channel Nurturing Intellect. Today in this video lesson you are going to study another important tool of recombinant DNA technology, that is the use of competent host for transfer of recombinant DNA. So let's begin. Now first of all, what does competent host mean? Competent host is the one which can take up recombinant DNA from the external medium. Now normal cells, they are unable to pick up foreign DNA because the DNA molecule which is there, it is hydrophilic in nature. So it is not able to cross the cell membrane of the cell. So in the first place, we have to make the cell competent so that it can easily take up the recombinant DNA from outside. There are different methods which are used for transfer of DNA into a host cell. Let's study them. First is chemical method or vector mediated method of gene transfer. So first understand the structure of the cell membrane and DNA before studying the method. So cell membrane, as we know, it is made up of lipid bilayer. So there are two layers of lipids present in the cell membrane. Now these lipid molecules, they are polar molecules. They have a hydrophilic head which is attracted towards the water and a hydrophobic tail. Okay. Now the head portion is having negative charge because of the phosphate group. So that's why you can see here in the picture that the outside of the cell membrane, it is negatively charged due to phosphates of the lipid molecule. Similarly, the layer of lipid which is towards the cytoplasm, its hydrophilic heads are facing the cytoplasm. The DNA molecule which is there, it is hydrophilic molecule and it is also having negative charge due to the presence of phosphate in its backbone. That's why the DNA molecule it is not attracted towards the cell membrane. So there is a repulsion. Okay, so these are the normal cells. That's why they can't pick up the foreign DNA from their external environment. Now, when we add or when we treat these host cell with a specific concentration of divalent cation like calcium. Okay, so these calcium, they neutralizes their negative charge and due to that, they increases the efficiency of the bacteria to take up the recombinant DNA by creating pores in their cell wall. We made the host cell competent by treating them with calcium chloride. To this solution, we add the recombinant plasmid and we incubate this solution on ice. Later, the solution is transferred to 42 degrees Celsius for 2 minutes. This is a brief period of heat shock and again we place them back on ice. So the competent cells, they take up the plasmid present in their surrounding and thus they get transformed. So this is one method, that is chemical method of transformation of host cell. That is, now let's study the second method, that is direct or vectorless method of gene transfer. In that, first is microinjection. In this method, the recombinant DNA is directly injected into the nucleus of an animal cell. So this method is used mainly for the animal cells. We hold the animal cell with the help of micropipette and then from the other side, with the help of microinjection, we inject the recombinant DNA directly into the nucleus of the animal cell. So here you can see this is micropipette. So this will prevent the cell from moving away and from the other side we are injecting the foreign DNA. The second method is biolistic or gene gun method. this method is suitable for plants and it is used for transfer of gene into the plant cell. In this case, what we do, we take microparticles of gold and tungsten and we coat them with DNA. Okay, so we take very small particles of gold and tungsten and we coat them with foreign DNA, and with the help of gene gun, we bombard the plant cell with high velocity these microparticles. So they get introduced into the nucleus of the plant cell. So this is another picture showing the biolistic gene gun method. Here you can see these are the microparticles which may be of gold or tungsten. We coat these microparticles with DNA. With the help of gene gun, we bombard the cells with these microparticles. So these particles, they get introduced into the nucleus where they deliver the gene to the cell. And method, that is the use of disarmed pathogen vectors. So when pathogenic vectors like TI plasmid and retroviruses, they are disarmed, that means their harmful genes are removed, and in place of that we introduce the foreign gene. So these disarmed recombinant vectors can be used for transfer of gene to a competent host cell. In our next class, we are going to study the method which is used to amplify the recombinant DNA, that is polymerase chain reaction. So stay connected. Like my video, please hit the like button, comment, share with your friends and subscribe to my channel. Thank you everyone.